And the first one is from the publisher. It's pretty short.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Now the other video, there's a link to it. I'm not going to play it here because it's a lot longer than I want to spend time on, but this is a good overview of meiosis, so I do strongly recommend that you click on this link in Blackboard so you can watch this video later. But we're not going to watch that one today. So the handouts, right, that we all worked on, yes? Does everyone see this?
Yes.
Okay, so one of the first things that we have to do is to replicate the chromosomes, right? Because what we're looking at here are 2 pairs of homologous chromosomes, but they do have to be replicated. And since I can't do color easily, that's why I just did the black, white, dots and stripes. Even though they're homologous chromosomes, I had to make them look different so we can see crossing over occur. So this is our first set, and I'll show a little bit of crossing over here, right between the adjacent sister chromatids. And we got the larger ones. Right, so we have to replicate first, and then I drew some crossing over. Remember, it's just between the adjacent sister chromatids. And you can draw some more crossing over. I just did a little bit to illustrate the process here. Right, so that's meiosis 1. So we're going to go through, and now we're going to separate our chromosomes. And remember, we separate the pairs, and we cut the number in half, right? So we'll say that this cell will get that one, and this cell gets the other one. Of course, they could have gone to the other cell, right? This is just one possibility. And then for the larger chromosome, right? So that's one possibility. But we could have, switched the smaller chromosomes and put them in the opposite cells, or switch the larger ones and put them in the opposite cells, right? Because it's random. Now, is it possible to have one daughter cell with the two little chromosomes and the other daughter cell with just the two big chromosomes? Is that possible? You got to talk to me. I can't see chats.
No, well, I guess if there was a mutation, but normally I would say no.
Well, normally, no. If it happens, there's something seriously wrong.
Yeah.
And if that gamete were actually to fertilize or be fertilized, then the resulting zygote is not going to be viable, right? So that should never happen unless there's a mistake. Now, like I said, we could have gotten, two cells. Maybe it has the small black chromosome and the larger striped one, and then a cell with the smaller, clear one or white one, and then the larger one with, dots, right? So there's more than one possibility. And so when it comes to how the different chromosomes end up in the two different cells, what do we call that? What is that process? What is that concept? Which guarantees a random mixture of the mommy and daddy chromosomes. What do we call that? Which guarantees a random mixture of mommy and daddy chromosomes. What do we call that?
Just variation?
Independent assortment.
Yep.
Ah.
Independent assortment. And when we get into genetics, we'll see this is called the law of independent assortment. So we've cut the chromosome number in half, right? But we still have our replicated chromosomes with sister chromatids. So meiosis 2 is just like mitosis, and we're just going to separate the sister chromatids. So I'll separate the little ones first. Right, so that's one possibility. It's not the only possibility, right? I could have mixed up the sister chromatids, right? I could have put this one over here and this one over here instead, right? So there's more than one possible combination because all the gametes are going to be unique, but there's more than one possible answer here. I've just given you one possibility. So does that make sense?
Hey, Professor, I had a question. So at the top level, when there's sister chromatids, is that the diploid cell, or is it the second level down that's a diploid cell? So the very first cell on top,
All right,
where
this
you have the
is
sister
a diploid cell.
chromatids.
At the end of meiosis 1, now they're haploid. So after meiosis 1, we have haploid daughter cells. We've cut the chromosome number in half. And then we're going to continue with haploid cells and then split the sister chromatids. So
I can't see what you're showing.
Yeah, go ahead.
I can't see what you're showing anymore.
Oh, you'll have to close that tab and come back.
Okay.
So this would work better if I could use colors more easily, but that's harder to do, especially in the original handout. You guys would just be sitting in class with a pen or pencil, so that's why I use different shading and markings, right, to show that. But if I'm going to say, for example, that in this homologous pair, let's say that this is chromosome number 3. We got number 3 that originally came from mom and number 3 that originally came from dad, right? So that's one homologous pair. And here we'll call this, chromosome number 17, and again 17 from mom and 17 from dad. So that's a homologous pair. It's the same chromosome, but remember, these are maternal and paternal chromosomes in you that came from your mother and your father. And so it's a homologous pair. You got two sets of all your chromosomes. Half came from your mother, half came from your father. So you got two sets, so you're diploid. Right, 2N. And then when you're haploid, you're just N, right? Because we've cut the total number of chromosomes in half. We still have our replicated sister chromatids, which we're going to separate in meiosis II. But all the gametes, again, they're all still haploid. And that way, when an egg and sperm come together, right, when two haploids come together, now you have a diploid zygote. So, any other questions on this? So this is just one possible outcome. There are, in fact, multiple outcomes here. And keep in mind that, remember, if we had those 23 pairs of chromosomes, those 46 chromosomes to start with, we would have over 8 million possible random combinations with just, 23 pairs. I'm just showing 2 pairs, so. So, any more questions on this, or meiosis in general? Okay, so now we're going to start talking about the molecular basis of inheritance. So now we're going to look more specifically at DNA, and we're going to learn about how they discovered that DNA was the genetic material. We're going to learn about DNA replication, and that will lead us to our next lectures on transcription and translation to see actually how the information that's stored in DNA is ultimately converted into a protein. So, life's operating instructions. So, of course, we now know that all the instructions needed to create a new organism are encoded by DNA. But it took a while for humans to discover that. In 1953, James Watson and Francis Crick, one was American, the other British, they shook the world. So they came up with the structure of a double-stranded helical DNA molecule. And of course, DNA stands for deoxyribonucleic acid, because the sugar is deoxyribose, as opposed to RNA, which is ribonucleic acid, because the sugar is regular ribose. So in 1953, the structure of DNA was determined. And so DNA, the most celebrated molecule of our time, because we finally were beginning to understand where the instructions were, and then how the instructions were being used to create a new organism. So DNA uses a chemical language which allows us to create new cells, new organism. And DNA itself is a relatively simple molecule, but the instructions are what are complex. And in terms of all the DNA that's in a cell, maybe half of it actually codes for proteins and the other half doesn't. But that doesn't mean it's junk. Many of those DNA sequences code for RNA molecules that help to regulate the expression of other genes. So the entire genome is complex. Although the molecule DNA itself is simple, what it does is very complex. And when we start getting into genetics, you're going to see that it's way more complicated than what Gregor Mendel, that monk that played with his pea plants, much more complex than he could ever have imagined. So early in the 20th century, so the early 1900s, we knew of the existence of DNA. It was discovered in cells because we could analyze cells chemically. We knew there were lots of proteins there composed of amino acids. And we knew there was this stuff called DNA, which is composed of 4 nucleotides, ATGC, right? Adenine, thymine, guanine, and cytosine. And they thought it was curious, but they thought that DNA was way too simple of a molecule to be used to store the information needed to create a cell, because DNA is a polymer of repeating subunits, A's, T's, G's, and C's. How could anything like that possibly result in the development of an entire new organism? It just seemed like a molecule that was too simplistic. Just 4 nucleotides. Proteins were a much more likely option for storing the information because proteins were made of 20 different amino acids. So proteins were much more complex than DNA. And so scientists first thought that, well, proteins must be the way that information is stored and used to create a new organism. But then why all this DNA? Why was all that DNA there? What purpose did it serve? And so some speculated maybe the DNA is actually the genetic material which carries the information. So they had to try and figure this out. So they did study bacteria and some of the viruses that infect them. And we'll look at one of the famous experiments that was done with that. But studying bacteria was a lot easier, than working with any other cell type, because bacteria were easy to grow, and you can grow millions very quickly and easily. It's easy to extract DNA from the bacteria by rupturing their cell walls. And so bacteria were the model organism they used in order to start looking for, the genetic material. Frederick Griffith, he studied a bacterium called Streptococcus pneumoniae. And it's one that can cause pneumonia in mammals. And there are 2 strains of Strep pneumoniae that he worked with. One that was pathogenic and the other non-pathogenic. And what does pathogenic mean? What's a pathogen? Well, no, not necessarily deadly. But what is a pathogen?
It's like a piece of bacteria or something that can cause disease.
Well, a disease-causing organism, right? So something that causes disease. The disease can be mild, right? Or it could be severe. It could be deadly, but not necessarily. And so he had these 2 strains of Streptococcus pneumoniae. So he performed 4 different experiments. And so let's see what kind of experiments he did. He used mice as his test animal because mice can be infected with Strep pneumoniae. And so the 2 strains that he used, the pathogenic strain made a structure called a capsule. A capsule is a thick layer of polysaccharides usually surrounding some bacterial cells. And the benefit to those bacterial cells is that it's hard for the immune system to attack and destroy bacteria surrounded by a capsule. So it's a defense mechanism. So the pathogenic strain had capsules. The non-pathogenic strain, no capsules. So his first experiment, he injected mice with live encapsulated strep pneumoniae bacteria. So these were bacteria making capsules, and the mice die. Right? So we just demonstrated that the strep with capsules kill. In the second experiment, mice were injected with live strep pneumoniae that did not make capsules, right? So these were the non-encapsulated strep pneumoniae bacteria, and the mice survive, right? So strep without, capsules. I'll just say no kill because I don't have room to write everything out. All right, so strep with capsules kill, strep without capsules do not kill. And that's because the capsule protects those bacteria. And so the immune system cannot fight them off, and the mice die. But if strep pneumoniae do not have capsules, then the mouse's immune system is able to kill those bacteria, so the mice survive. So next experiment, Griffith took some of the strep pneumoniae bacteria with capsules and heated them up to kill them, so they were dead. And he injected the dead strep with capsules into the mice, and the mice survived. No big surprise because the bacteria were dead, right? So dead strep with capsules. And again, we'll just say no kill. Right? So those first three experiments, they demonstrated that live strep with capsules kill. Live strep without capsules do not kill. Dead strep with capsules do not kill. Okay? So the fourth experiment, what Griffith did is he took the live strep without capsules and the dead strep with capsules, mixed them together and injected them into the mice, and the mice died. They were dead. And what Griffith found reproducing inside the mice were live strep with capsules. But that's not what the mice were injected with, right? They were injected with dead strep with capsules, which we know do not kill. Live strep without capsules, which we know do not kill. So these two things do not kill. But when Griffith mixed them together and injected them into the mice, then they killed. So how can two things that normally do not kill, when you mix them together, then they killed? And the organism that was recovered from the mice was not the one they were injected with, right? The organism recovered was strep pneumoniae with capsules, living strep pneumoniae with capsules that were reproducing inside the mice. So do we follow what's going on here? Right, two things that normally do not kill, you mix them together and now they kill. So what was going on here? Well, what Griffith surmised was that somehow there was something from the dead strep with capsules that was transferred to the live strep without capsules. And so Griffith called this a process transformation, something that was heritable, right, able to be inherited, was transferred from the dead bacteria to the live ones. And the live ones got transformed and started to produce a capsule. And so this suggested that there was something heritable inside these bacteria that was able to be transferred to other bacterial cells. And we do know this process, and we call it transformation. It is a natural process whereby bacteria can absorb short linear pieces of DNA from the environment. And so what happened here is that the dead strep with capsules, they were dead, so they broke apart, right? They decomposed. Their chromosomes broke apart. And there were little pieces of DNA floating around. One of those pieces of DNA contained the gene to make a capsule. And so one of the live strep, without capsules, absorbed that gene, right, to make a capsule. And then they started making a capsule, and the mouse's immune system was not able to kill those bacteria. So they reproduced, and they killed the mice instead. So transformation is a natural process whereby bacteria can absorb short linear pieces of DNA from the environment, and in that way they can acquire a new characteristic. So that's was something, kind of important that they discovered, although they didn't know exactly what was going on with the DNA yet. That was later determined. But so bacteria do not have to evolve a characteristic through successive mutations. They can simply acquire it instantaneously, from another species, and they've got that characteristic. So lucky them. But what this showed is that there was something inside the bacteria that was heritable, that could be inherited and was transmitted from the dead bacteria to the live bacteria. And so those live bacteria acquired the characteristic of making a capsule. And we call that transformation. In a future lab, we'll talk about transformation, because we do this in lab as part of a recombinant DNA technology, where we clone genes into bacterial cells. And it's very easy to do. It's ridiculously easy to do. Although we don't use short linear pieces in lab, we use small circular pieces called plasmids. And because the bacteria do not normally absorb plasmids, we're going to force them to absorb the plasmids, which again is very easy to do in the lab. But we now had some evidence suggesting, okay, we know there's something inside the cells that can be transferred to other cells. But what is that something? Is it DNA? Is it protein? Now, of course, we know it's DNA, but back then they didn't know yet. But any questions on these 4 experiments and what they mean? So again, now we do know that it is possible for bacteria to acquire genes from the environment, and so the bacteria can change their genotype and therefore their phenotype, what they look like, by simply acquiring genes from the environment. So some more evidence that DNA was most likely the genetic material, again using bacteria, but also the viruses that infect bacteria. So every living cellular organism has some kind of virus that can infect it. So every living thing on Earth, there's some kind of virus that can infect it, including bacteria. And we call those kinds of viruses bacteriophages. So a bacteriophage, the word literally means bacteria eaters, although the viruses are not eating the bacteria, they're using them as factories to produce more viruses, eventually killing that host bacterial cell. But bacteriophages have a protein coat called a capsid. Inside the capsid is the chromosome, a piece of DNA. And what we're seeing here in this picture is that the bacteriophages, which are kind of scary looking, they land on a bacterial cell and they attach to that host cell using cell surface receptors, and that's what all viruses do. But here, the bacteriophage uses an enzyme, lysozyme, to, I always use the expression to drill a hole through the cell wall, but they use that enzyme to basically eat a hole through the bacterial cell wall. And then the whole capsid structure, it contracts like a syringe and squirts the DNA inside the bacterial cell. And we see the DNA here being squirted inside. So now this bacterial cell has become infected. And the bacterial DNA will take over and force that bacterial cell to make more viruses and kill that bacterial cell. But they didn't know that back then, right? So they didn't know what all was going on inside that bacterial cell. But they knew there were bacterial viruses which could infect and kill bacterial cells. And so the question is, okay, we know the bacterial virus has a lot of protein. It has that capsid, right, that protein coat. It's also got a piece of DNA inside. Which one is causing the infection? Which one will result in the formation of new viruses? Is it in the protein or is it in the DNA? So they used a phage called a T2 bacteriophage. It's one of the so-called T-even bacteriophages, T2, T4, T6. It's just a particular type of bacterial virus. And that's the one that they use because these viruses infect E. coli. So it's very easy to grow E. coli in a lab. So again, they used the bacteria because they were easy model organisms to work with. So what they did, they used radioactivity in this experiment. So in one experiment, they inoculated a flask of E. coli with the T-even bacteriophages, and the flask contained radioactive sulfur. Sulfur is an element that is found in amino acids like cysteine. So sulfur is only found in proteins, not in DNA. And so they let the bacteriophages infect, and then they separated the phages from the bacteria by putting them in a blender just to remove the phages from the surface of the bacteria. And then they centrifuge that mixture. So when you centrifuge, you're spinning, at very high speeds, and it pulls, heavier things down to the bottom. And in this case, the bacterial cells are much heavier than the bacteriophages. And so the bacteria cells will be pulled down to the bottom of the tube and form what's called a pellet. So solid matter is the pellet. In the liquid were the phages, because they're, way too light, right? So they stay suspended. They are not pulled down to the bottom. And then they looked for the radiation. Where was the radioactive sulfur? If the protein coat, if the capsid was the mechanism of genetic information, then we should find the radioactive sulfur inside the bacteria, right? Because the proteins would be taking over the bacterial cell. But they didn't find any radioactivity in the pellet. So no radioactivity was in the bacteria. It was still in the bacteriophages. It was still in the capsid proteins. So that suggested that, okay, it's probably not the proteins, responsible for the genetic information, which would cause the infection. But to be sure, they did a similar experiment only this time they used radioactive phosphorus. Amino acids do not contain phosphorus. DNA does, right? The phosphates are used to join nucleotides together. So there's a lot of phosphorus in the DNA molecule. And so they grew the bacteria and the phages in the radioactive phosphorus. Again, they used a blender to separate phages from the bacterial cells, and they centrifuged it, so they got a pellet and a liquid, and they looked for the radioactivity now. Now the phosphorus was in the pellet, not the phages. So this suggested it was the DNA from the bacteriophage which got inside of the bacterial cells and then would force those bacterial cells to make more bacterial viruses. And so this experiment by Hershey and Chase strongly suggested that DNA was the genetic information because it was being transferred from the bacteriophage to the bacteria. And so that suggests that's how the infection occurs. It's caused by the DNA. Therefore, DNA must be the genetic material. And so this was pretty good evidence suggesting it's DNA, not protein, that is the genetic material. So, of course, we know that DNA is a polymer made up of just 4 nucleotides, adenine, thymine, cytosine, and guanine. So that's our nitrogenous base, our A, T, Gs, and Cs, a sugar, deoxyribose, and a phosphate group. So chemically, we had determined what DNA was made out of. But how did all those things come together to form a DNA molecule? Erwin Chargaff was another scientist who, again, was trying to help determine that DNA was the genetic material. And so he analyzed the base composition of DNA, right, the percentage of adenines and thymines and cytosines and guanines. And he did this for different organisms, and he discovered that every different species has a different percentage of cytosine, guanine, thymine, adenine. So the fact that all these different species have different compositions of all those nucleotides, that suggests that DNA must serve an important purpose. Otherwise, why wouldn't we all just have the same amount of everything in our DNA? We don't, right? There are different amounts of cytosine, guanine, and thymine, and adenine. And that suggested that DNA served an important purpose. Possibly it was the genetic material. Now, Chargaff also determined the percentage of guanine equaled the percentage of cytosine. And then the percentage of adenine equaled the percentage of thymine. At the time, he did not really make that association between base pairing because he hadn't yet determined the structure of the DNA molecule. But Watson and Crick later used this information, right? The fact that there were the same number of G's and C's and the same number of A's and T's, right, to help determine the base pairing for the DNA molecule. But we had lots and lots of evidence now suggesting that DNA was in fact the genetic material of the cell. So it did take a while to convince biologists that this simple molecule, of repeating subunits, of repeating A's, T's, G's, and C's, was in fact the molecule responsible for storing all the information needed to create a new organism. And then the challenge was to determine how the structure could account for its role in inheritance. So how can this simple molecule store the information? So 2 more scientists, Maurice Wilkins and Rosalind Franklin, they used a technique called X-ray crystallography as a way to help determine the structure of molecules. And there was some controversy here because Rosalind Franklin, and her team, she was the lead scientist, I think, she did produce these images, and we see one of them here, which Watson and Crick used without her knowledge. And so they used her research to help determine the structure of a DNA molecule. Watson and Crick won the Nobel Prize. Rosalind Franklin was barely even mentioned, at least back then. Later, she was given all of the acknowledgments that she should have gotten back then. Unfortunately, she was dead. She only lived to age 37. She died of ovarian cancer, I think, which may have, at least in part, been caused by the X-ray exposure, the techniques they were using. But, she was not happy at the time because her research data was used without her permission, and then she barely got any acknowledgement at all, at least not officially. Only later did she get all the acknowledgments that she deserved. But Watson and Crick deduced that DNA was a double helix, and they used those X-ray crystallographic images to help determine that. And then with Chargaff's data on the percentages of G's and C's and A's and T's, they helped determine the base pairing. And for that, they got a Nobel Prize. But Rosalind Franklin, she had already concluded that DNA was composed of two anti-parallel sugar phosphate backbones with nitrogenous bases paired in the molecule's interior. So she already had a general idea of the structure. But again, she didn't get any of the credit for that. And of course, we do know that adenines are paired with thymines and cytosines are paired with guanines. So here we see an example. Now, notice that the phosphate backbones are used to join all the different nucleotides together. We have our nitrogenous base and the deoxyribose sugar and then the phosphate. Now, what I want you to notice here is that when you label the carbons on the ribose sugar, and we're not going to worry about that, but you give them numbers. And so you have different numbers on the carbons. And so that gives the molecule a directionality. And so you notice how, and this doesn't look quite right here because of the way that it converted, but we have what's called the 3' hydroxyl group on this end here. And then on this end, it's the 5'. That little tick mark is prime. And you'll notice that this strand runs 5 to 3' this way, but the opposite strand runs 5' to 3' in the opposite direction. So the two DNA strands run anti-parallel, right, to each other. So there is a directionality to each strand on the DNA molecule. And that's something that'll be important when we get to DNA replication. Now notice that in between the nucleotides, they're base paired using hydrogen bonds. And we know a lot about hydrogen bonds, right? We know that hydrogen bonds are weak bonds, right? Water molecules constantly make them and break them, right? They're very weak bonds. But there is strength in numbers. When a piece of DNA or a chromosome is made up of thousands or hundreds of thousands of nucleotides, they're sharing, many, a huge number of hydrogen bonds. And that keeps the two strands together. And so that double-stranded nature of DNA is one of the things that helps to stabilize it. Right? So DNA, when it's double-stranded, is a fairly stable molecule. And so you need a stable molecule in order to store genetic information that you're going to use to create a new you. Now, RNA is also extremely important, but as we'll see, RNA is nowhere near as stable as DNA. RNA is just single-stranded, and RNA has A's and C's and G's, but no T. It's replaced with U, which stands for uracil. Uracil is not as chemically stable as thymine. So they actually believe that RNA evolved first. And there actually may have been primitive RNA cells called ribocytes or ribocells, which use RNA as their initial genetic information. But RNA really not stable enough. And so eventually that led to the evolution of DNA, a much more stable molecule. The thymine was replaced, or the uracil was replaced with thymine, which is more chemically stable. And the DNA molecule is double-stranded, which makes it even more stable. Now, you can see the number of hydrogen bonds shared between the A's and T's and the G's and C's. Right, so how many hydrogen bonds are shared between the A's and T's?
2,
And the G's and C's?
3.
3, right? So, but that's what allows for the base pairing, is the hydrogen bonding. All right, so adenine and thymine can form 2 hydrogen bonds, and cytosine and guanine can form 3 hydrogen bonds. So Watson and Crick were able to help determine the specificity of the base pairing, also including the data from Chargaff about the percentages of A's and T's and G's and C's, so it made sense. So we see the hydrogen bonds being formed. Now the hydrogen bonds, like I said, they're weak bonds, but they're strength in numbers, and they keep the double strands together, which makes it more stable. But hydrogen bonds can be relatively easily broken by the cell using enzymes, because the cell needs to pull those 2 strands apart in order to replicate and make more DNA, or to transcribe and make RNA. So there is strength in numbers, hydrogen bonds, but they're not so strong that the cell cannot pull them apart when the cell needs to. Now, the relationship between structure and function is manifest. And manifest is a fancy word that means clear or obvious. Well, I guess if you're a scientist and you were working on DNA and you came up with that double helical structure, you might say that. For all the rest of us, we need to have it explained to us before we can understand why the DNA molecule is the way it is. But back then they thought it was kind of, well, you know, a eureka moment. Well, of course it has to be like this, right? Well, okay for them, but for the rest of us, we have to understand how everything comes together in order to form that double helix. Now, each of the 2 strands of DNA will serve as a template for the synthesis of new DNA, right? Because we know that DNA gets replicated a lot. It gets replicated before mitosis, it gets replicated before meiosis. So each strand will serve as a template, and the new DNA that's synthesized is called a complementary DNA strand. And so the cell will separate the double-stranded molecule. Each strand serves as a template for the synthesis of complementary DNA, A's with T's and G's with C's. Right, so that's how the base pairing goes. And then we have our replicated DNA. So when it comes to base pairing, and you will have to write me some complementary DNA. If I gave you the sequence, what would be the complementary DNA sequence here?
CTAATGT.
All right, so remember, A's with T's and G's with C's. So that's the complementary base pairing. So before we get into the mechanism of DNA replication, we have to talk about some experiments that were performed to help determine how the replication works in terms of producing the new DNA molecules. Is it a mixture of half old original DNA, half brand new DNA? Is it all brand new? So there were several models or several hypotheses about replication and the end product. One of the hypotheses was the conservative model. So if we look at the first diagram here, we have our parent DNA strands. If they go through replication, the idea here is we still have the original parent double-stranded molecule, and then we'd have this new, brand new double-stranded DNA molecule where both strands were brand new. And then if you have two brand new strands, they'll yield more brand new DNA, and the old original DNA will yield a brand new DNA, plus the old original double-stranded molecule is still there. It's still conserved. So this was one possible hypothesis. Another hypothesis was the semi-conservative model, where each strand serves as a template to synthesize new DNA. And so the quote-unquote new DNA molecule is actually half old original DNA and half brand new DNA. So it's semi-conservative, right? So half of the old original DNA strand is conserved, it's still there, and then a brand new complementary strand is synthesized. In the dispersive model, the quote-unquote new DNA molecules are actually a mixture of old and new DNA on each strand. It's kind of hard to see here, but the idea with the light blue and dark blue is that each strand is a mixture of light blue and dark blue. And when you have more cycles of replication, the old original DNA, the dark blue, gets less and less. And there's more and more new DNA, the light blue. And so there's less and less of the old original DNA and more of the new DNA. So these were three hypotheses, right? Conservative, semi-conservative, or dispersive. And so the experiment to determine which one was correct was performed by Messelson and Stahl. And so here they used two types of nitrogen, because the A's, T's, G's, and C's are nitrogenous bases, so they've got lots of nitrogen in them. And so the two forms of nitrogen were regular nitrogen, N14, and then a heavy isotope of nitrogen, N15. And remember that isotopes have extra neutrons, so that makes them heavier. So again, they use bacteria, easy for bacteria to reproduce, and then to study their DNA. So they first grew the bacteria in a culture medium containing the heavy nitrogen. And that's meant to be the dark blue is the heavy nitrogen, and the light blue is the lighter nitrogen. So at first, they were grown in the heavy nitrogen. And then they were transferred to a nutrient solution with regular nitrogen, right, the N14. And they let them go through at least 2 replication cycles. And then they would extract the DNA, and they would centrifuge it using cesium chloride. So cesium chloride, CeCl, is a salt. And it's a heavy metal type salt, right? Cesium. And you can use cesium chloride to separate DNA by weight. And so when you put your DNA into the cesium chloride and you centrifuge it at very high speed, it will separate out DNA based on weight differences. So we'll look at the incorporation of heavy nitrogen versus light nitrogen to help determine which model was correct, right? Conservative, semi-conservative, or dispersive. Now, it is interesting to note that the blue line here representing the DNA, it's not arbitrarily blue. The authors chose blue colors for a reason. And the reason why, when you centrifuge the DNA, it forms a blue band. That's the actual intrinsic color of DNA. It's blue. And you can see that in the cesium chloride because the cesium chloride is white. And you see this blue band. So the blue color is actually what the DNA looks like in the cesium chloride. It's actually blue. But remember, dark blue is the heavier nitrogen, light blue is the lighter nitrogen. And by centrifuging it in cesium chloride, you can separate DNA by weight. So here are the possible results. So if the conservative model is correct, if the old original double-stranded DNA molecule is conserved, if it's still there after replication, then we'll have an original heavy nitrogen, a double-stranded DNA molecule, and then a brand new light nitrogen DNA molecule. But both strands are brand new and they're lighter. And so when you centrifuge this in the cesium chloride, you'll get the heavier, right, conserved band, and then a lighter, right, band of all brand new DNA. That's not seen. If you did a second replication, you would still expect the original double-stranded molecule to be there if it's conserved. Remember, it was made first in the heavy nitrogen. All new DNA would be made with the lighter nitrogen. And we would have seen two bands, a heavy band and a light band, right? So the same bands, and that is not observed, right? So you don't see that. If the semi-conservative model is correct, then when you have the first replication cycle, one of the strands will be the old original heavy DNA, and the other strand will be the lighter new DNA. And so each double-stranded molecule is half old, half new. And because we're using the heavy nitrogen initially, and then the lighter nitrogen later, we'll see a mixture of DNA strands, half heavy, half light together. And that gives us an intermediate band in the middle. So neither heavy nor light, it's intermediate because the DNA molecules are half light DNA, half heavy DNA. And then if we take this DNA and let it go through another replication cycle, we'll again have half old heavy, half new light. But when the lighter strand is used as a template, it synthesizes a light strand with that light strand. So we have all light. And we would see two bands, right? The intermediate band, right, are half old heavy, half new light DNA, and then a band of all light DNA. And that's in fact what we see, right? So here we've already given the evidence which says that replication is semi-conservative, but if the dispersive model had been correct, with the first cycle of replication, we would also have had an intermediate band, just like the semi-conservative model, because in the dispersive model, each strand of DNA is now a mixture of old and new DNA. so heavy and light DNA, but that's in each strand. And so we get an intermediate band, which looks just like the semi-conservative band. But if we go through another replication cycle, if the dispersive model had been correct, that original heavy DNA, it gets less and less, right? Because the new DNA is being synthesized with the light nitrogen. And so over time, there's less and less of the old original heavy DNA and more of the new light DNA. And so we would get a different intermediate bands forming. And we did not see that. So it is the semi-conservative model, but they used a DNA that was replicated using heavy nitrogen first, and then it was replicated using the light nitrogen. And then the different weights of DNA were separated in the cesium chloride tube. And so it's just an experiment that was done to show that replication is semi-conservative. The original DNA strand that was used as a template is still there and part of the double-stranded molecule, but the complementary DNA strand is brand new. So half old, half new, semi-conservative. So does that make sense? So when it comes to replication, it's complicated. It is a process that requires multiple enzymes and multiple proteins in order to happen. Now, when it comes to the speed with replication, it's pretty quick and accurate. A DNA polymerase, the main enzyme for synthesizing DNA, it can add nucleotides at a rate of about 1,000 nucleotides per second. So every single second, 1,000 nucleotides are added, 1,000 ATGCs in 1 second. So it's incredibly fast. But chromosomes are really big. And in order for a cell to be able to replicate and divide relatively quickly, a cell needs to be able to copy the DNA quickly. And I think I mentioned this before, but bacteria, they only have 1 circular chromosome, so they can replicate that and divide in as little as every 20 minutes, whereas one of your cells requires almost a day in order to replicate and divide. So it takes a lot longer for all those long linear chromosomes. So when it comes to talking about DNA replication, every textbook uses bacterial DNA replication as the model of how it happens in all cells. And the reason why we use the bacterial model is that bacteria, most of them have just 3 polymerases, DNA polymerase 1, 2, and 3. And so that's what we talk about. Eukaryotes have up to 15 different DNA polymerases. So it's much more complicated to talk about those. And so most textbooks don't, right? They just talk about bacterial DNA replication. So just keep in mind that what I am describing to you is what happens specifically in bacteria, which is basically the same thing that happens in eukaryotes. It's just that DNA polymerase 1, 2, and 3 are bacterial polymerases, not eukaryotic polymerases. And that's something I think that students don't really, get when they learn this, because they learn about DNA polymerase 1, 2, and 3, but they don't understand that's just in bacteria. Eukaryotes have different polymerases. Most of them are labeled with the Greek alphabet, alpha, beta, gamma, whatever polymerases. And so it's much more complicated. So what we're actually focusing on is how it actually happens in a cell, but specifically we're talking about the enzymes in bacteria, not eukaryotes, but the same idea. And so look at all these possible enzymes and proteins. DNA helicase, which unwinds the DNA, right? The DNA is double-stranded and it's helical, It has to be pulled apart and unwound in order for a DNA polymerase to actually make more DNA. We have clamp proteins, binding proteins to stabilize that double-stranded molecule when you open it up and pull it apart. Topoisomerase, which deals with the winding problem. A DNA gyrase specific to bacteria, we'll mention it here, and that's it. DNA ligase, the word ligate is a verb that means to join together. And then primase is an enzyme that adds primers, which are very important that we'll get to. And then telomerase, we'll see that and what it does. And so we are going to go a little bit longer today for lectures, so we're going to go into the lab time to make sure we get through this, because lab is not all that long today. And this is where I wish we could be in the classroom, but you've all played with rubber bands, right? You know what a rubber band is? So if you take the rubber band and you use your index finger and thumb and you hold one end, and then use your index finger and thumb on the other hand to hold the other end, and then you start twisting it in opposite directions, right? So that we are simulating our double-stranded DNA molecule with a rubber band. And so you twist and twist and twist, and then if you kind of move your hands together, what happens to that rubber band? Right, so take the rubber band, hold it between your thumb and index finger on both hands and start twisting in opposite directions, and then kind of move your hands toward each other. What happens to that rubber band?
The twist isn't tight anymore.
Oh no, it is. It actually bunches up on itself, right? If you twist and twist and twist, and then you move your hands together, that rubber band then bunches up on itself. It really gets twisted all on itself. And that's called a torsional stress. You know, torsion, T-O-R-S-I-O-N. And this is something that the cell has to deal with because the DNA molecule is a double helix, right? So it's kind of curly. And so there's some stress there, right? We'll call it twist stress or torsional stress. And so when you pull the strands apart, that really increases the stress in front of where you're pulling it apart, right? It makes the twists a lot tighter. And so that creates an interesting physics problem, right? Because the physics of DNA replication is pretty amazing when you think of what the cell has to deal with this curly double-stranded molecule. It's got a lot of torsional stress. And when you pull it apart, you increase that torsional stress even more. And so we have topoisomerase. Oh, the red ones are the ones that I'm really talking about right here. They're kind of important. And so topoisomerase is an enzyme that helps to relieve that extra twisting stress. It actually does it by nicking, right, by breaking one of the bonds between the nucleotides on one of the strands so that it can rotate about the other strand freely to relieve that tension, right, to relieve that, you know, twisty stress. But the point that I want to make is that it's a complicated process in terms of the physics behind it, with all this twisting and all the stress caused by that twisting. And so cells have to deal with that in order to replicate the DNA. So when it comes to where replication begins on a DNA molecule, there are places called origins of replication. That's where the cell is going to start pulling the two DNA strands apart. In a bacterial cell, which has one circular chromosome, there's only one origin of replication. But eukaryotes have linear chromosomes, and they're long, right? Hundreds of thousands, millions of base pairs long. They're huge. And so there can't be just one origin. That would take forever. So when it comes to eukaryotes and linear chromosomes, there are many origins of replication, many places where the double strands will be pulled apart and allow replication to proceed. And replication will occur bidirectionally. So bidirectional replication, you see these red arrows here, right? So when you pull the strands apart, replication goes this way and replication goes that way. And in that way, a eukaryotic cell can replicate those long linear chromosomes relatively quickly. Not as quick as bacteria because they just have one circular chromosome, but many origins of replication. And you end up with your two daughter DNA molecules, half old original DNA and half brand new DNA. So here we're seeing one of the problems with DNA polymerase. Remember that our strands run 5' to 3' in one direction and 3' to 5' in the other direction, right? So the 3' end had that hydroxyl group. And so there's a 3' hydroxyl group. And so a new nucleotide can be added onto that 3' hydroxyl group. So if we're replicating DNA and we're adding to the 3' end, what that means is that replication goes from 5' to 3', right? Because we're adding onto the 3' end, so we're growing in that direction, 5' to 3'. My little circles are meant to indicate nucleotides. Right, so DNA can only add nucleotides to the 3' end. And so DNA can only be synthesized in one direction, 5' to 3'. And this creates a problem because each strand of the DNA molecule is going to be used, right, to synthesize DNA. So if we separate our strands, if this strand goes 5' to 3', this strand goes 5' to 3' this way. So if we're synthesizing new DNA and DNA can only be synthesized in one direction, and I don't know if I can. So if we're synthesizing new DNA here, and new DNA here, we have a problem, right? Because we can only synthesize in one direction. Of this complementary strand, it goes 5' to 3'. That's the right direction, right? But what about this strand here? If it were going as shown, it would be 3' to 5'. And that doesn't work that way. So when the upper template DNA strand is used, then DNA replication would be going the wrong direction, 3' to 5'. When the lower DNA strand is used, in this example, then DNA replication goes the proper direction, 5' to 3'. So what do we do with this one? It's going the wrong direction. DNA polymerase can only synthesize new DNA in the 5' to 3' direction. So one of the template strands is all good. New DNA will be synthesized 5' to 3' here. So that's all good. But what about here? It would have to go 3 to 5. That's the wrong direction. It can't do that. Right, so DNA polymerase can only add nucleotides to the 3' end, which means the growing DNA strand is growing in the 5' to 3' direction, not the opposite way. In bacteria, the enzyme that adds nucleotides is DNA polymerase 3, and it will synthesize one of the new DNA strands as one long piece, one continuous piece. We call that the leading strand. But the other new DNA is in the wrong orientation. It will be synthesized, but in little pieces called Okazaki fragments that we'll see. So the leading strand is synthesized continuously as one long piece in the 5' to 3' direction, but the lagging strand is synthesized in little pieces called Okazaki fragments. Now, the pieces that are synthesized will be done in the right direction, 5 to 3, but that requires some gymnastics for the DNA to perform. And because all these little fragments are synthesized as pieces, they are not continuous, right? There are little gaps in between, and they'll be filled in by DNA ligase, right? Because it joins DNA together. So, do we all know what railroad tracks look like? Have we all seen trains? Right, we know about railroad tracks. And so a railroad track has how many rails? It's got 2 rails, right? Well, DNA has 2 strands. And so think of the DNA molecule as a railroad track. And so the enzyme DNA polymerase is like the train on that track. And so DNA polymerase needs a piece that's double-stranded in order to hop on and start to replicate to make more DNA. The DNA molecule has been pulled apart. So now they are individual strands, they're single strands. A train can't hop onto a railroad track with only 1 rail. Yes, I know there are monorails. We're not worried about that. So the idea here is that there needs to be a piece of DNA that's double-stranded in order for the DNA polymerase enzyme to hop on. But when you pull the DNA apart, now you've got single strands. And so that's a problem. And that problem is solved by the enzyme primase, which adds primers. A primer is a little piece of DNA or RNA. In this case, it's going to be mostly RNA, which is added, and that creates a region that's double-stranded so the DNA polymerase can hop on. And so let's just have a DNA strand here, and we have a little primer here. Our DNA polymerase enzyme can hop on to double-stranded region. As it's synthesizing new DNA, it creates the second rail as it goes. So it adds those nucleotides and creates the second rail so that it can keep on moving. And so the polymerase enzyme makes that second rail so it can keep on moving, but initially to get started, it's got to hop on to double-stranded DNA. And that's accomplished by primers. On the lagging strand, the one with all the Okazaki fragments, all the primers are RNA, not DNA. Right? They're RNA. Right? There's no RNA in DNA except during its initial synthesis. And there's actually some RNA in DNA. So We need the primers in order to allow DNA polymerase to hop on and start to replicate. As DNA polymerase is replicating, it's creating, right, the second rail. It's creating that second strand. So it keeps on moving. But the Okazaki fragments, it's a problem. And so here we're seeing a diagram to illustrate. We have a piece of DNA. It's single-stranded because it's been pulled apart. DNA, the enzyme, the primase enzyme, adds an RNA primer and creates a region that's double-stranded that allows DNA polymerase to hop on and begin to replicate and start synthesizing that new complementary strand in the correct direction, 5' to 3'. When the polymerase reaches the next RNA primer, it stops, and then another DNA polymerase enzyme, DNA polymerase 1, in bacteria, it will cut out the primer and replace it with DNA. And then DNA ligase will seal everything together. So the lagging strand with all the Okazaki fragments and all the RNA primers, all the RNA is going to be removed and replaced with DNA. All the little gaps in between, all the fragments will be sealed together and it will be made whole. It will be made complete. But it's more complicated for that lagging strand to be synthesized than the leading strand. So again, to summarize, a lot of the enzymes used, helicase will unwind the DNA. It opens it up and pulls the strands apart. Then there are single-stranded binding proteins to stabilize that open DNA molecule. And when the two strands are pulled apart, it creates a lot of extra twisting in front where the strands are being pulled apart. So topoisomerase helps to relieve that extra twist, that extra tension. And then primase adds the RNA primers. The DNA polymerase 3 in bacteria is the main enzyme that adds nucleotides. The DNA polymerase 1 will remove the primers and replace it with DNA. DNA polymerase 2, which is not seen here, is a DNA repair enzyme to repair mistakes that happen later. And then DNA ligase will join all the fragments together and make them whole. So a lot of enzymes and proteins are required. And so here is a common diagram shown in just about every introductory biology textbook, and it's wrong. It's just meant to illustrate all the parts and pieces that are required in order for DNA to be replicated, but it's wrong. But here we see our leading strand is being synthesized 5' to 3' as one long continuous strand. And then here we have the Okazaki fragments on the lagging strand, and they're being synthesized in little pieces. They will be synthesized in the 5' to 3' direction, but it's not possible the way the DNA molecule is shown here. What actually happens, and we'll watch a beautiful animation of this, is that this strand loops around. And when it loops around, it's now put in the proper orientation for DNA synthesis to occur in the 5' to 3' direction. A better diagram, one that you don't need to know, but I just added this to help illustrate the idea, we see how the lagging strand is looping around. And so this is a much more accurate diagram than this one is. And so that lagging strand, right, the strand that's going in the wrong direction, loops around and orients itself so that DNA can be synthesized in the proper 5' to 3' direction. But it's more complicated than what this shows. And it seems kind of confusing how when you pull the two strands apart, and then you have to make one of them loop around in order to be in the correct orientation. But this more accurately represents what is going on inside of a cell. And like I said, we'll see the animation next. So the various proteins that all participate actually form a large protein complex. So a DNA replication machine, which is called the replisome. So it's a combination of polymerase and helicase and topoisomerase all there together at the same time to make this work, right? To make replication happen. And what they have observed, it looks as though the replisome complex is actually stationary and the DNA moves through it. So rather than the polymerase enzyme actually moving across the DNA, it looks as though the DNA molecule is actually moving through the replisome complex. So let's go back and see if I can. Oh, I'm going to have to open this link. So give me a second to go back and get it to open it up. Yes, let me get there. I hate YouTube. All right, so let me share my screen so we can watch this. Let me turn down my volume a little bit.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
And that's so good, we're going to watch it again.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
And of course, we know the DNA molecules are not brand new, right? They're half old original DNA and half brand new DNA. But we get to see an excellent example, an animation of how the whole process works. Now I got to get back to where I was. And it does happen quickly. Remember, up to 1,000 nucleotides every single second. So all those enzymes are working together to pull the DNA apart, unwind it, relieve the excess tension, the excess stress that's in the molecule, add all those nucleotides, and a DNA polymerase does this with remarkable accuracy. So a few mistakes do happen, and a DNA polymerase will correct those mistakes as it goes along. Now, DNA polymerase is not 100% perfect, right? And actually, we do not want DNA polymerase to be 100% perfect. We need mistakes to happen. We need mutations to happen. Why?
So the fittest can survive, like evolution and stuff.
Right, so we need some mutations, right, to allow for variation and adaptation. So DNA polymerase is not 100% perfect, and we do not want it to be. Yes, many mutations are bad, but you got to take the bad with the good, otherwise there's no good, right? There's no adaptation. And so, but DNA polymerase does do a pretty good job, but every once in a great while it makes a mistake that does not get corrected. But we do have other DNA repair enzymes to correct mistakes that happen. So just sort of a FYI thing here, prokaryotes can have up to 5 different DNA polymerase enzymes. Most commonly, it's DNA polymerase 1, 2, and 3. 3 is the main one that adds the nucleotides. DNA polymerase 1 removes the RNA primers and replaces it with DNA. And DNA polymerase 2 is a repair enzyme, right, to repair mistakes that happen later. And a lot of mistakes do happen. You know, some estimates up to 1 million lesions occur per cell per day in the eukaryotic genome. So that sounds like a lot. In reality, it's just a tiny fraction of 1% of all of the nucleotides in that cell. But still, 1 million, problems every single day, if they're not corrected, that will add up very quickly. And that organism will die if it doesn't correct these mistakes. So one of the mistakes is something that occurs with ultraviolet radiation, so UV light. UV light can excite 2 adjacent thymine nucleotides and cause them to chemically bond to each other. So if you have 2 thymines side by side, UV light can stimulate them so they bond to each other, and they form a thymine dimer. The problem with a thymine dimer, it does not look like 2 thymines to a DNA polymerase enzyme. And so when DNA polymerase comes along and it hits that thymine dimer, either the polymerase falls off and stops replicating, or it adds the wrong nucleotide on the complementary strand of DNA. It does not add 2 adenines. It makes a mistake, right, because of that thymine dimer. Now, we have DNA repair enzymes, all forms of life that evolved being exposed to the sun has these DNA repair enzymes, because UV radiation comes from the sun and it causes damage, right, to the DNA of organisms exposed to that UV radiation. And so we have these DNA repair enzymes constantly scanning the DNA. If they find a thymine dimer, they cut it out and replace it with 2 proper thymines. And so this happens all the time. But if you've ever, gotten too much sun, if you got sunburned, your skin turns red, indicating you've damaged your skin cells. And if you got a really bad sunburn, you blister. And the reason why you blister is because you've killed those skin cells. You caused so many thymine dimers that they could not be repaired. Lots of mutations occurred, right, when the cells were trying to replicate and divide, or when the RNA polymerase was trying to transcribe, right, to make the RNA to make proteins later. That didn't happen. And so those skin cells died. That's not a good thing. And so too much UV exposure leads to cumulative damage because not all the thymine dimers get repaired. And that damage accumulates over time and can lead to skin cancer, right, to melanoma. But we do need some UV exposure. Why? Why is some UV beneficial?
Vitamin D.
Right. It causes the synthesis of vitamin D in our skin. So we do need some skin exposure, some sun exposure. But, if you have lighter skin, you don't need as much sun exposure, maybe 10 to 15 minutes on your hands and face, and you make all the vitamin D you need for that day. If your skin is darker, you need more sun exposure in order to make the same amount of vitamin D. But too much UV causes damage, and we can actually use UV light to sterilize surfaces, right? Because the UV kills the bacteria because it creates so many thymine dimers they can't replicate, they can't divide, so they die. But we all have these DNA repair enzymes. Well, I say all. A tiny, tiny fraction of humans have rare genetic disorders like xeroderma pigmentosum. And so those poor individuals who are unfortunate enough to be born with that mutation, if they are exposed to sunlight for like 5 seconds, they get sunburned because they have no ability whatsoever to repair the thymine dimers. Those individuals can never go out during the daytime unless they are completely covered from head to toe to block UV. They can only go out at night after the sun has gone down. And back before the creation of LED lights, incandescent bulbs, they make UV. Fluorescent bulbs make UV. So these people couldn't even be in a room with an ordinary light bulb unless that light bulb was shielded. So they had to use a lot of candles, oil lamps for light, right, because those don't produce any UV. So this is a really horrible condition. Most people, if they have it, only live until their 30s, and they usually die of skin cancer because exposure to UV is almost impossible to avoid. And those people do not have any of the repair enzymes needed to replace thymine dimers. So a very rare condition, fortunately, but a very bad one as well. So when it comes to linear chromosomes, there is yet another problem. When DNA replication occurs, it's not possible to replicate the ends of a chromosome faithfully. And so because a linear molecule, the polymerase enzyme is only going to be able to replicate one of the strands, not the other one, because we're at the end of the chromosome. So every time your cells go through mitosis and cell division, your chromosomes get a little bit shorter, right? Because that's a problem, right? You don't want your chromosomes to get shorter, but that's what happens with linear chromosomes. With a circular chromosome, like in bacteria, this is not an issue. It's circular, right? There's no loss. But with a linear chromosome, the ends get shorter. And it's just a consequence of replication. It's not possible to replicate all the way to the very end of linear chromosomes. And so they get shorter each time. And that's the problem. So what evolution did is we have extra strands, or not extra strands, but extra DNA composed of repeat sequences on the ends of your chromosomes. So they don't code for anything. They're not part of a gene. And so every time they get shorter, the genes on the very ends of your chromosomes are protected because the telomeres are there to protect them. So those end repeating DNA sequences are called the telomeres on the ends of your chromosomes. And they do get shorter with each cycle of mitosis and cell division. But they are there to protect those genes on the end. But at some point, with enough cycles of replication, all the telomeres are gone. And now you have reached those genes on the ends of the chromosome. And as soon as those start getting shorter, well, those genes are not going to produce a functional product anymore. And so the cell is going to age and die. And this is what happens with eukaryotes and their long linear chromosomes. They get shorter every time. But the telomeres, those extra sequences of DNA, are there to protect the genes. And the telomeres get shorter and shorter and shorter, and eventually the gene starts to get shorter and shorter. At that point, it's all over for that cell. Now, there is an enzyme called a telomerase, which repairs the telomeres. So every time the replication cycle happens and the telomeres get shorter, the telomerase will go and elongate them. It'll repair them. And so, well, that sounds great, except that a telomerase is not produced in all cells all the time. It's an enzyme that is produced in the gametes, in the sperm cells, because you don't want those new chromosomes to be shorter, to start with when you fertilize an egg. And so the telomeres get protected. They get recreated by telomerase. We also find telomerase in fetal cells, because remember, that fertilized egg, that zygote, is going to go through a lot of mitosis and cell division to create a fetus composed of billions of cells. So that's a lot of replication cycles. And so the enzyme telomerase is active during fetal development. And that protects your chromosomes so that you won't, terminate once you become a fetus. We also find telomerase being produced in adult stem cells, in skin cells, because you're constantly replacing, right, your skin cells. So telomerase is there. It's also in your T and B lymphocytes, which are very important immune cells, which go through a lot of mitosis and cell division when they're fighting a pathogen, so they're protected. And also cancer cells. They produce a telomerase. And that's why cancer cells are considered to be immortal cells. They will live forever. If they have the proper environment and proper nutrition, a cancer cell will just continue to divide and divide and divide forever, because its telomeres are protected by telomerase. Most of the rest of the cells in your body, no telomerase. And that's actually kind of a good thing. It doesn't seem like it, but most of the somatic cells, so everything that's not eggs and sperm, and not stem cells, and not skin cells, and not T cells or B cells, so basically 99% of everything else, they have a finite number of cell divisions, because each time the cells replicate and divide, the telomeres get shorter. Eventually, all the telomeres are gone, and now those genes on the very end will start to get shorter, and those genes will become non-functional. So the number of cell divisions is called the Hayflick limit. So it's the number of times a cell can divide before it dies. And the ranges vary depending upon source, either 40 to 60 replication cycles, cell divisions, or 50 to 70. But the point is, there is a finite number to the number of times most of your cells can go through mitosis and cell division before they die. And it's because the telomeres are getting shorter and shorter. Because of that, it's estimated the maximum possible lifespan for human beings would only be about 125 years. And that's assuming everything else is perfect. You are not born with any genetic conditions. You eat the healthiest diet. You exercise. You don't get excessive sun exposure. You don't smoke. You don't drink. You don't do drugs. You get enough sleep. Everything else is perfect. And then it may be possible to live up to 125 years old. But that's only if everything else is perfect. So some scientists have said, well, if we can turn on the gene for telomerase and all those other cells, then it can just keep on dividing forever, which means that we could live forever, right? So do you think it's possible that telomerase could lead to immortality? And would that be a good thing?
Cancer cells would still have telomerase.
Well, yes, and if you activated the telomerase gene in all your other cells, what's to stop them from becoming cancer cells?
Yeah, like cancer would still kill people off.
Right, so that would be a problem. But most people say, oh yeah, I'd like to live a lot longer. But would you really? I mean, think about how many people are depressed right now. Are they going to be more or less depressed 1,000 years from now or 10,000 years from now? I mean, some of you have a hard time getting out of bed as it is. What are you going to do 1,000 years from now? Death is what gives life meaning, what makes us want to live. It's because we know we're going to die. So immortality, not likely. And even if it's possible, I don't think people would be happier if they lived a whole lot longer. Because look at how unhappy people are right now. So telomerase, it's a nice idea to play with, but I don't think it's really going to work. And I don't think it would be a good thing anyway. So any comments you want to share